Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. This time on Careers in Discovery, I'm with Remi-Martin Laberge, co-founder and chief technology officer at Arda Therapeutics. Remi and I talked about his journey from academic research to building companies, the lessons he picked up along the way, and how Arda is using single-cell sequencing and targeted antibodies to eliminate disease-driving cells in chronic conditions. We also discussed the leap from scientist to founder, applying oncology tools beyond cancer, and why curiosity and risk-taking go hand in hand in biotech. I am back on Careers in Discovery, this time with Remi-Martin Laberge of Arda Therapeutics. Remi, welcome to the show.
Thanks so much for having me, Tom. This is great to be here.
Fantastic to see you. And I always start by talking about what you're up to now. And I'm fascinated by Arda Therapeutics and the work that you're doing there. So tell us a little bit to start with about eliminating pathogenic cells and the approach that you're taking to treating disease through that.
Yeah, happy to do that. So what we do at Arda Therapeutics is we find pathogenic cells in chronic diseases. And the intent here is to get rid of those cells for therapeutic benefit, right? So the approach that we're taking is a single-cell RNA-seq approach where that helps us to identify those bad cells, right? And once we find those bad cells, we look for cell surface receptor that are enriched in those cells and to build antibodies, right? Antibodies that we weaponize, and after weaponization, these antibodies find the cells and get rid of the cells. Mm-hmm. So think of us as a company that is using oncology tool, but outside of oncology, right, to treat chronic diseases. So that's pretty much what we're doing. We think this approach, it's definitely differentiating versus what other people are doing, right? But it's also a more holistic approach in the sense that instead of alleviating the pathways that these bad cells are engaged in and leading to pathogenicity, we think that getting rid of the cells altogether may have enhanced therapeutic benefit. And the reason is simple, right? So if you affect a pathway, you have redundancy in the cell system, and you may lead to resistance, or you may be just incompletely affecting the bad aspect of these cells, where if you get rid of these cells totally, you will have the maximum effect you can have, so long as you can identify those bad cells from the get-go.
Yes, I see. Okay. And I would assume, and this is an assumption, so correct me if I'm wrong, that there's an element to this which in chronic conditions, these pathogenic cells accumulate, right?
That's right. So what we find, right, to take a simple example of fibrotic diseases, which is one of the main indications we are working on. So in the context of fibrosis, what happened is you have injury into the microenvironment in the tissue, and then that activates a wound healing response, right? So you have some cells of fibroblasts that are differentiating to help with wound healing. They will start producing collagen, for instance. And that collagen is beneficial in the context of wound healing because it is there to make sure that you have a scaffold and tissue integrity remains normal. But when collagens accumulates, it starts cross-linking. Yes. And when it cross-links, what happens is it forms a scar. Similar to a scar you would have in the skin during the wound healing. That also happens in many other organs upon wound healing if that wound doesn't get resolved. And that's the problem, right? So in some diseases where you have fibrosis, lung fibrosis, liver fibrosis, other type of fibrosis, is what happened is you have the accumulation of these cells and they continue to spit out those collagens. The wound never resolves, and that leads to loss of tissue function because of scarring of the tissue. Yes. So that's an example. So these cells accumulate. They normally should resolve if the wound healing has resolved, but in some contexts, in the context of disease, they remain there. So it's 1 + 1 = 2. Get rid of the cells, and you will prevent right? That further accumulation of collagen,
Yes.
and therefore you should reduce progression of disease.
Yeah, it's fascinating. And, you touched on this earlier, we've developed all these novel tools in pursuit of cancer treatments, right? And it was only going to be a matter of time before they started to be applied elsewhere. Obviously, cancer's been the big catalyst for a lot of this advance, but a cancer cell is just a type of cell, right? So why should you not be able to use them elsewhere?
Yeah, definitely. Yeah. So that's the idea. Cancer cells are cells that are becoming dysregulated, and that accumulates. And we've developed these tools to get rid of these cells. The venue of chemotherapy originally was essentially to take advantage of the rapid progression of cancer cells and creating poison into the cell cycles, things like that, to get rid of the cells. Now, come the 90s and the early 2000s, with the avenues of antibodies and more specialized treatment, now that we realize that, okay, we can affect on the cell surface of these cells nodes that are essential for progressions that cancer cells have become addicted to. And now, fast forward a few years later, now you have T cells engagers, antibody drug conjugate, where essentially, in the case of T cells engager, you're doing a T cell that is normally going to be there to kill other cells, and you kind of connect them to the cancer cells to get rid of those cells, right? So you do that, and that works very well. T cells engager, analogous to the CAR T, the same idea that you affect the T cells directly affect the cells, or antibody drug conjugate, which is an approach we are taking at Arda, where the antibody will bind to the cell of interest to eliminate them, but you weaponize this antibody by attaching to that antibody a cytotoxic payload. Upon internalization, the cytotoxic payload gets released into cells, and you have a specific killing of that cells that have, for which the antibody have bounded to.
Yes, I see. Interesting. And you've been with the company from the beginning, and you've been working with them over the last few years to advance the therapy.
So the company got, I think, incorporated at the end of 2021. So my co-founder, Adam Friend, started at the company, and then, I can tell you of the origin of how this
That'd be great.
But I joined right away after we incorporated it, and we've been in operation since the beginning of 2022, and I was leading research there for a while, and now the team has grown quite a bit. And so it's been almost 3.5 years now. Time's fly.
Yeah, it does. No, I'd love to hear the origin story. I always find it fascinating to understand because what I typically see is a pattern of there's lots of interesting conversations, and then suddenly something happens that sparks the actual decision to start a company. And really interested to hear about where that happened for Arda.
Yeah, so maybe I'll intersect my career path with Adam's path and how we coalesced again to start a company. So for me, I joined a company called Unity Biotechnology at the end of my postdoc, and so at Unity, what we were doing, and the company is still going, so we were eliminating pathogenic cells as well, but a specific subtype of pathogenic cells that are called senescent cells. So these cells accumulate also at sites of chronic diseases, and they are essentially tumor suppressor mechanisms. Cells that are damaged, they start producing in a microenvironment. It's called senescence-associated secretory phenotype. And the idea there was, well, if we get rid of those senescent cells, we might have therapeutic benefit. So the approach of Unity is analogous to what we do at Arda, but it's kind of flip on its head. Instead of finding the pathogenic cells first, we postulate that at Unity that the senescent cells are there at chronic disease. So let's find things that will kill senescent cells and see if there is therapeutic benefit in those diseases. Which is different than what Arda, we say, okay, let's forget about senescent cells. Let's find pathogenic cells and then build a tool against these ones, right? So we've been doing that for a while at Unity with a few clinical trials, osteoarthritis. Now there is ocular clinical trials that are still ongoing that just revealed recently. And the idea was let's get rid of those bad cells, but specific bad cells, senescent cells. But I've always thought that, okay, senescent cells is a subset of pathogenic cells, and we're looking under the lamppost by targeting directly those senescent cells. What if there are other pathogenic cells that, if we remove the constraint of cellular senescence, would also lead to therapeutic benefit? So that's something that we didn't have the bandwidth at Unity to pursue that angle, but always had in the back of my mind.
Yes.
Now, in parallel, my co-founder Adam Freund was working at Calico, and Calico is a company it's an age-related company that's sponsored, that got started by Google, and he was a PI there. So I was a PI at Unity, and Adam and I know each other from when he did his PhD in the Campisi lab, and I did my postdoc in the Campisi lab. So we've known each other for many years, right? Yeah. With broad interest in aging, age-related diseases. So he went to do his career path, I went to do mine. We remained friends, and we chat over and over again, over beer sometime, but also just chatting here and there. And so he was the first one who said, look, I want to, and COVID always helping, like the COVID crisis of, all right, so what am I doing with my life?
Yeah.
Is this really what I want to do, right? And I have this idea for innovation, and I feel a little bit constrained in the box in the company that I'm working on. So Adam decided, like, oh, that's it. I'm going to start my company, right? And so Adam started this pitch deck, and we've exchanged ideas about how to do that outside of senescence, and really put together all of the ideas and how he would be doing that, right? And I was peripherally involved at the time. I was, reviewing his pitch deck. I'm sure other people were doing that as well, but he was kind of consulting with me, given my experience of eliminating pathogenic cells, specifically in the context of cellular senescence. And so then he incorporated the company and found some seed money from A16Z plus a couple of other VCs, and at that point said, okay, Remi, do you want to join to lead research, right? You know how to eliminate cells, you know how to establish the right readout, you could help with indication selection, so do you want to join? And then I joined as a co-founder. And so that's essentially how we started the company, but really that was, Adam taking the plunge and say, look, let's get rid of bad cells. And this could have, it could have happened 10 years ago, but not in the same way that we could do it like 5 or 6 years ago. And that is because of single cell RNA-seq. The key piece here is identification of those pathogenic cells, right? And single cell RNA-seq allows you to look in a diseased tissue and identify subsets of cells, right, that have evolved characteristics that are pathogenic. Now, that doesn't give you causality just yet, but you can have a strong association with something is causal to disease, for instance, collagen production in the context of fibrotic disease, but also what are these cells being enriched in the disease, right? So now you have kind of a two angle, and then at the middle of that Venn diagram, now you can identify those pathogenic cells. That still doesn't tell you the causality relationship, but you're getting close and very close. And now if you can find that these cells are also showing up in preclinical model, for instance, then you can go test the hypothesis of elimination of these cells would lead to therapeutic benefit. So that was at the early onset of the company. This was the approach. It's still the approach now. We added a bunch of bells and whistles to refine the identification of those pathogenic cells, but that is the key for the company.
It's really interesting, and it struck me that one of the key insights there is that senescent cells are just a type of pathogenic cell. Or a subset of that, and it's not even really a leap of logic, right? It's a simplification almost, but that unlocks a lot of other things.
Exactly. So senescent cells may contribute to some disease, right? And I think they're accumulating evidence that this is the case. But most of the cells we're finding now by looking at pathogenicity are not senescent cells.
Right.
So this was really the point of the iceberg, essentially, those pathogenic cells and those senescent cells. There is way more than that. And I think this is what now, shifting a little bit the approach to identify pathogenic cells first and try to eliminate them rather than looking for senescent cells and postulate that they will have pathogenicity it's flipping this on its head has helped us tremendously to unveil a bunch of other cells that are not necessarily senescent.
Yeah, fascinating. I could talk to you about this all day, Remy, and I often say this in the podcast because I have such interesting conversations. But I want to talk about you specifically and your career journey and sort of how you ended up in sort of aging research and how you ended up taking the path that you were in. Particularly because, and we've kind of given the narrative, but of course, you were involved in aging research is very sort of high profile right now, but you were involved in it before that was the case, and so keen to talk about that. But I'm always interested in the early days of people's careers and sort of, for you, why science? Why biology? Why sort of this route in the first place?
Yeah, I mean, look, I guess as many scientists have always been curious to understand how the world around me works, right? I've always been interested in medicine as well. So biology was always close to me, but physics as well, chemistry, big data. I really love mathematics, right? During my teenage years. And I've always took a Socratic approach to life, right? And asking the why and why and why, right? And maybe sometime bothering my friends and people around me is like, okay, so you think you know that, but really, what's the evidence? Why? How is this working, right? And and so that led me into science without knowing too much what science was, right? As a teenager, but I always knew that this was something I wanted to do, and my interest in mathematics and all of that, so it just led me into natural science, and then eventually into in college having to decide what I would do, like chemistry, biochemistry, biology, or whatever. So I chose biochemistry, not knowing exactly what I would getting into. Yeah. And I have to say that I've had mixed feelings during my undergrad on my career path and decisions. And the reason is that I don't like to learn things by heart, but I'm very good at, retaining something if I understand it first. Right. So it always starts with the curiosity, right? I'm curious about something. Oh, I'm gonna dig very deep into it, and I will learn all of what's needed around it, right? Just by connecting the dots, right? And that was my undergrad, and said, all right, so things that are interesting me. And after an undergrad in biochemistry, you can go work or you can continue to study, and I've never really liked working, so I continued to study, right? So went to do a master degree and then a PhD. And this is really where the spark hit, right? Now being in the lab, postulating hypothesis and go test it. And that was extremely satisfying to the questions that I've always asked myself is, how do we know what we know, right? Is, you know what you know because you isolate a variable in the lab and you go test that hypothesis. And isolating variables, it's easy, quote unquote, in math, right? Because it's a pure system. In biology is messy. Yeah. So you try to repeat an experiment, same system, and you have different results. So you need to repeat multiple times, and then you look at the average of that, and then statistics gets involved. And it's, to me, was really exciting. Yeah. It's not exciting for everyone, but okay, so you need to dig into a system, understand it very well, understand the parameters, what are the boundaries of your readouts, where does it start, where does it stop, right? And what is your dynamic range of the assays? And very, very technical type of questions. Oh, this is how we can conclude on biology and within the confidence interval of the results. And that to me was a revelation, right? And then digging into it. And at that point, my PhD, I was working on oncology in drug resistance, right? So I kind of learned a little bit of in vitro pharmacology there, not necessarily being taught that, right? Pharmacology was not a field of study that I've had been exposed to much, but starting doing it just by trial and error, right? And understanding what are the limitations there. And then I continued to want to work in oncology, and so this was at my PhD was at McGill University in Montreal, and then I moved to California to join the Campisi lab, and Judith Campisi, who passed away last year, unfortunately, was working on cellular senescence, right?
Yeah.
And what Judy has uncovered during her many years, one of the things she has uncovered is that senescent cells in proximity to cancer cells kind of fuel the cancer cell to grow faster. And I was quite interested in digging into this aspect. So I joined the Campisi lab not to work on the senescent cells themselves, but more on the adjacent cells, the cancer cells that are next to it. So I proposed that to Judy, said, all right, yeah, join my lab, come do a postdoc, started a project, and they said, well, can you work on this in the side, right? And so I started working on other projects in the side, and I got really interested now in okay, what are those senescent cells doing? And this is how I got exposed to aging research. I was not interested in aging research that much before, right? But my life has always been like, okay, let's do puzzle solving, right? And at some point you realize that, oh, there might be application to what you're doing, and that's very exciting. So the Campisi lab was at the Buck Institute for Research in Aging in California, and it's fantastic environment to be exposed to aging research in general. There are all sorts of type of aging research, right? And using different model organism from yeast to worm, flies, mouse, and age-related diseases. And it's a microcosm that is somewhat unique, and it's very, very exciting. And so I started working on that, working on multiple projects, very exciting time. And until I find out that, you can actually modulate a couple of nodes in senescent cells and get them to apoptose, right? So what I found is that if you get rid of the anti-apoptotic process, right? If you block the anti-apoptotic upregulation in these cells, they directly change their fate from senescence to apoptosis. So these were the first senolytics observation, right? And that led to the company formation of Unity Biotechnology. So originally, I was solving puzzle. This was all exciting. My path was, I'm going to become a professor at a university somewhere, writing grants, doing my research and all of that. And then Judy Campisi started this company, Unity Biotechnology. And I was in the middle of creating essentially the data package that led to that company formation, plus a bunch of other people, right, from the Van Dersen lab, the Kirkling lab, and so on. And so I said, well, we need someone to lead the discovery of new senolytics. Do you want to join? And now that was my, after deciding to do a PhD and a postdoc, now that was really a fork for me of deciding, am I pursuing the academic path or do I join a company? So at that point, the decision was kind of hard because I'd already set my mind to, I'm going to be an academic and run my own lab. But what was appealing to me is a little bit what I was saying is, solving puzzle is fun, but realizing that you can have therapeutic potential and real life application was really appealing, and I've never really considered it this way before. That was one thing. The second thing is, what did I want to do in an academic career was to start my lab and working on cellular senescence, identifying senolytic pathway, and see what is the impact on chronic diseases. This was exactly what I was meant to do, but in industry, with the bonus of not having to write any grants. So it took a bit, and then essentially I accepted to join the company. And this was a big transition for me now, from academia to essentially industry. But in a sense, the first few years at Unity Biotechnology was more applied, more pharmacology driven. But this is what I would have probably done in my first years, at least in academia. So I was pretty happy compared to doing that. And then I stayed 8, 9 years at Unity Biotech after. Yeah, and then there was the crisis of COVID.
Of course.
Yeah, and I wanted to go back to Canada at that time.
Ah, I see. Okay.
Yeah, so there was some fire in California. There
Mm-hmm.
was the pandemic. It was apocalypse, right?
The
And
end
I
of the
said,
world, yeah.
all right, yeah, exactly. I was not seeing my family in Canada. I said, all right, let's move back to Canada. So I found a job that I kept for 1 year. Um, at the University of Montreal, but it affiliated with the University of Montreal. Really, it was an independent um, endeavor sponsored by the government that aim to identify academic projects and company startup to bring them from the research side of things into company creation or licensing opportunity. So think of it as a VC firm that is nonprofit and Canadian government sponsor, right?
Okay.
And that entity is called IRICOR. So I was the head of scientific operation there, portfolio of about 20 projects, discussing with academic, helping them align in their drug discovery projects, and then interfacing with my business development partner, with company, right, to see if there would licensing opportunity, intellectual property opportunity, and making sure that those are being protected. So that was good. I did that for a year, all remotely, right? The intent was to move back to Montreal. But I felt that was a little further away from the actual research, and I missed that, right? And so this is when Adam started Arda, and had the idea, and was kind of bouncing idea off of me. And then at some point, this coalesced into, look, do you want to join the company? And I said, yeah, right, let's do it, right? So I kind of quit IRICOR at the time, and then to start with Arda.
Yeah, and I suppose by that point the post-COVID norms had become a bit more norm, right? And
yeah, exactly. Yeah. Like the fires in California were behind us. Well, to come back, but anyway.
Sure. Yes.
And then COVID was the shock of COVID was less strong. I mean, I still would like the family to be closer, but hey, that's what it is.
I'd not thought about it like that from a California point of view, actually. And that you have this sort of global disaster of COVID, but also like the world around you is literally on fire, and it must be a surreal situation.
Yeah, there was, and it keeps coming back, right? And it's a challenge for sure in California. It has been for many years, and it come and goes in terms of the intensity. But in Northern California, so I live in the San Francisco Bay Area, and that particular summer, the first summer of COVID, was particularly bad. Yeah. There were a couple of days where, at noon, you could not see the sun. Right. So this was really apocalyptic. And because of all the light, it's all of the particles in the air, right? It turns the sky red in the same way that it does at dusk, right, and at dawn. And so the sky was red, right, at noon. And you have COVID, no one in this outside, and you have obviously a lot of smoke outside, right? So it's hard to breathe. You would probably, if it was not for COVID, you would nonetheless wear a mask because the air is just. And this was kind of a surreal summer, I have to say.
I can imagine. And it makes you pine for Canada for sure.
yeah, exactly. I mean, although a year later there was giant fire as well in the northern part of Quebec province, and the wind just carried all of that smoke into the New York area. Yeah. Anyway, so where there's forest, there will be fire for sure.
Yes, exactly. I was also interested, sort of as you were talking about your path to this. Clearly, you're someone who's been driven by finding a really interesting problem and then figuring out, as you talked about, figuring out all the parts that surround that problem and then looking at different ways you can solve it. I suppose, the shift into industry then is an interesting one. I think, at Unity, I can understand, you weren't running the company, you were a researcher in the company, and so that you were probably a bit shielded from that. You're probably able to get on with your research a bit more than maybe you are today. But drug development, of course, is quite a focused pursuit, right? There's the initial exploratory stage, but then you've got to pick something and you've got to back it and you've got to go with it. And I imagine there are a lot of people who have a similar mindset to you in that you want to pursue your curiosity. How do you balance those things, do you think?
Yeah, that's interesting, right? So there, I always say that, in academia, you run experiments, and in industry, eventually you run assays, right?
Right.
An assay needs to be reproducible and all of that. Not that it's not reproducible in academia, but the impetus is less there in academia, right? So you write your paper, then you're done, and hopefully it can be reproduced by someone else. Most of the time it is, but not always, right? In industry, the rigor of the assay development has to be there if you want to compare molecules to each other, because you cannot always put all the same molecules in the same assay, right? Yes. But you need to balance that with the innovation. You cannot always have perfect assays all the time. At some point, you need to have a quick experiment that will answer the question, right? And so, yeah, you need the right balance, and you need to make sure that you understand when it is time to run something that's rigorous versus something that is good enough, right?
Yes.
And that's the nuance that I think you get. And how do I balance now? Once you have all of enough pieces of the puzzle in the early research side to say, okay, that's interesting enough. I think we've had enough hits that let's move things forward. Let's build a lead molecule, and let's cross all the T's, and dot all the I's on these molecules. I think there is the excitation and the plus the anxiety of things reproducing, that I think keeps it interesting to some extent, right? If the first time you get your great results, say, all right, now let's put it to the test. Let's redo the experiment, like in vivo, especially, right? Does that work again? Does that work similarly? Yes. Is it context dependent, or does it work no matter what, right? And this becomes very interesting. And how do I balance that curiosity of the early days with the rest? As I mentioned, I'm very interested in mathematics and data science as well. And pharmacology is also a big puzzle, right? Right, yes. Because you have pharmacokinetic. What is your minimum efficacious dose? What is your maximum tolerated dose? How are you going to come with your first in-human dose, right? What is your therapeutic index? So this is a mathematical problem and challenge. And that keeps it interesting to me. It's very complex to match the pharmacokinetic that you will have in a mouse, right? And making sure that it's going to work in people as well. Yes. And okay, we're using antibodies. The antibody may not behave exactly the same. Is your antibody binding the same way to your target? Do you have expression of that target in another organ in your preclinical model that may create a sink for your antibody? And therefore, when you look in the blood, you have a reduction of this antibody. So, all right, what does it do to? Oh, that kidney express a little bit of that target. Yeah, but that's irrelevant for people because it's not expressed in people. Okay, so what does that going to do for efficacy? So you see, that there is always a puzzle to be solved. Yeah, and to me, that's what keeps it interesting. And of course, at some point also is making sure that you're surrounding yourself with the right people. I am not a toxicologist. I am not a pharmacologist, in fact, right? So I understand enough of all of those pieces to know what questions are interesting to be asked, but I'm not an expert. So you need to surround yourself with experts that do know what they're doing more than I do, but also that have that keen interest for their field of study, right? I'm very passionate about what I'm doing. I want people that are passionate about what they are doing, and I think it's key. That's the way to go, right? Drug discovery is really much a team effort.
Yeah, absolutely. And if you're surrounding yourself with those experts, you've always got the opportunity to continue to learn, but continue to learn in relation to something that you remain very interested in and very engaged with. Yeah, it makes total sense. So tell me a bit about the last 3 years, Remy, and sort of building the company. Obviously, you'd seen a startup at Unity, and you'd been on that journey with them. And I think seeing the first 8 years of a project is a long time, right? It's you see a lot of shifting there. But now you 2022, you're in an environment where now you've got to build a company, and you've got to build a team, and you've got to raise money, and you've got to do all the other stuff that comes with that. But I guess tell me a bit about that journey. Tell me a bit about what's been surprising, what you've I'm just interested in what that last 3 years has shown you.
Yeah, so that's interesting. So I think we've had a few phases, I'd say, at Arda, right? So when we had the seed money, which was about $7.5 million, right? And what Adam and I were thinking is, look, with this money, let's get to proof of concept. Proof of concept that we can identify bad cells, right? Well, let's find the right indication. Let's identify the bad cells. Let's make tools to eliminate them in a preclinical model. And once we have that done, then we're going to raise our Series A, right? And so we got to that point, and the financing environment got a little challenging when we got to that point. So it was a little harder to find the Series A money. The data looked good, but it was a little harder, and it's just because of the timing, right?
Yeah.
So we got that, and then what happened is eventually we did get that Series A money, but along the way there was something very, very serendipitously good that happened to the company. Is we hired our CSO, right? So Scott Turner, who was at Pliant Therapeutics, was the CSO of Pliant Therapeutics, an expert in fibrotic disease. I've developed small molecules that made it into the clinic, and so we heard that he became available. So we had a couple of discussions with him, and I think that helped with. So when we hired Scott, that helped giving confidence into investors that not only we will be able to do the proof of concept of eliminating the cells, and especially that we're interested in fibrotic disease, that hiring Scott gave a little bit additional credibility that we would be able to take those findings and bring them to the next level, right? And back to your question earlier of, to do the transition from early research into the drug development, and Scott's credential helped. So when we hired Scott, around the same time, we started getting more traction with VCs, and that helped tremendously. So this is kind of, I'd say, Arda maybe 2.0, where we had a second phase of expansion with Scott coming on board, and then the Series A money. So now the company is 17 people, I think, 18 people. And there's still quite a bit of early research that's happening. We have our lead molecules that are moving forward into our main indications, and it's pretty exciting. And it's all about the team, right?
Yes.
It's having the right people at the right place, doing the right thing at the right time, right? Yeah. And it's Scott and I responsibility to make sure that this occurs on the R&D side, and things are looking great. In terms of getting money in, and investors relationship and business development and things like that, Adam takes the larger part there, right, as the CEO, and of course, Scott and I are always there by his side, and we had a lot of great discussion. There was JP Morgan in January, where we've met with people. People are interested in what we're doing. on the platform side, but also on the potential drug discovery side, right, in the advanced program. So, we have good tractions. We're doing rigorous science, and that's all starts there, right? If the data at the end of the day looks good and it's reproducible, you'll be successful.
Yeah, absolutely. And obviously, the market agreed, and in a challenging time, you're able to do a really good Series A raise. It's interesting, a couple of things you talk about there, I think, because, fundraising is obviously a hot topic for a lot of people at the moment and a lot of companies. You're doing something novel, right? There's proxy in that these technologies work in cancer, but the way you're applying them is novel. And that has some benefits from a investor attraction point of view, because it's not the same as everything else. At the same time, they have to be willing to take a bet on something novel that doesn't have that track record, right? And then the other part was that, so interesting in your thoughts on that. But the other part was sort of, you talked about Scott there, and when I look at biotech companies, I always think, well, I don't know enough, and this would be the same for investors. I don't know enough to say that this is great science, but this guy does, and he's on board, and so. It, like you say, it's that credibility, that track record, but also that sort of confidence that, okay, he's done it. He knows what to do. He knows the pitfalls. I guess both of those things are critical. But yeah, just interested in your thoughts on those.
Yeah, definitely. So on the investor side, to your first question about how risky it is to use oncology technologies outside of oncology. Yeah, that's part of the appeal. That's also part of the risk, right? Will this work? And there is really two things there. The first one is the therapeutic hypothesis, elimination of the cells leading to therapeutic benefit, which we can de-risk in preclinical model, but then, it's always in clinical trials, really, that you'll know. So that's a bet on that angle. The other angle is the potential toxicity. Obviously, some of those tools that are used in oncology comes with toxicity risk and known toxicity as well. Yeah. And you can afford those toxicity in oncology because the alternative is worse, right? For that reason, the early indications we have selected are also happen to have really high morbidity profile.
Right.
Like, for instance, idiopathic pulmonary fibrosis has a prognostic, right, between 3.5 to 5 years in terms of life expectancy upon diagnostics, which is ballpark what you'll get in oncology as well, which allows us to take a little bit of a risk in the toxicity side because the alternative is worse than not taking those risks.
Right.
And so that allows us to go the risk a little bit technology in an indication for which otherwise the outcome is worse. But we want to expand beyond that, so we'll need to we have to be careful on the type of tools we're using and the type of toxicity profile of the modalities we're using. Antibody drug conjugate versus T-cell engagers versus other means of cell elimination. And in terms of the credibility and for the investors, that's a good point. So, I think, I'm not a VC person, but clearly, right, there is a little bit of an appeal for VCs to invest into a funder that has done it before. Yeah. Or to sometimes different models of VCs, they will put a management team that has done it before, or they bring their own management team, right? And that gives them confidence that at least the operation will look good. Now, as for scientists bringing into the company someone who has done it before brings credibility. And exactly to your point of, right, if the VC might not know enough about a topic to determine if the science is good, the idea may sound exciting, right? But is the science good? But hiring the guy that will be rigorous enough and knows about the science, and if that person is willing to join, well, it's probably good, right? And so that gives that credibility for sure. Yeah. And that I think, hiring Scott, I think, gave that signals to our investors. Now, it's the gestalt, right? It's one piece of the puzzle, but that was one piece that I think was helpful.
Yeah, and like you say, you've got to have really compelling science in the first place, or you don't get that guy on board, right? Like
That's right. That's right.
Yeah, it makes sense. So thinking back on the journey with Arda and with Unity and with the year in Canada and looking back on where you've come from and where you've been, if you bumped into yourself now, Remi, perhaps back at the Buck Institute, in the early days of your work there, what do you think you would share? What advice do you think you would give, or what's the one thing that you think you've learned that, from a career perspective, that has been most important?
Yeah, I would probably tell myself to not be afraid to take risk, right?
Mm-hmm.
And I think that's important for anyone in science, maybe in life in general. I mean, depending on the type of risk, obviously. But take a chance. Continue to explore. And I've always done that, the exploration and all of that. But I've always been a little bit conservative with the risk aspect until I plunge, right? So maybe I would have plunged earlier into those type of things. Not that I would change anything. I mean, I'm saying this, but I'm going to contradict myself. I would not change anything because I believe I'm where I am. I'm happy where I am. And all of my previous experience led me to this, right? So if I change something, maybe that would be worse. So I'm happy with where I am. I would not change anything. But don't be afraid. Don't be anxious to take those risks. And I think that's important, right? Because we tend to be comfortable, especially in academia, unfortunately, it takes so much time to go through the process that at some point you maybe start being comfortable in what you're doing, and that's not fantastic, right? I think if you're exploring multiple things in your life, right, in science and in fields, you'll have a better understanding of maybe what interests you even more, right? And than what you're currently doing. And to me, the best way to excel at something is to do something you like, because you will put the hours and it won't be a chore, right? So if you do what you like, you'll be successful at what you're doing, and you'll be happy because you're doing what you like, right? So I mean, it's a full circle there. But you cannot find what you're very good at and what you really like until you try many things. So take those risks, go explore, right? Be curious. Curiosity kills the cat. Curiosity builds the scientist.
Yeah, okay. I like that. I might steal that. Fab. So we've talked a lot about Arda and what you're working on there. Can you tell us a bit about, where you are today, what you're excited about, or, what's next?
Yeah, so, we raised our array in the fall, so we're maybe 6 to 8 months in now. And, now it's about execution, right? We have the plan, we have the team, we know what we're doing, we know how to do it, and the data just need to cooperate. And so let's move forward. So we have a couple of programs that are in lead optimization. We want to move those forward, and we want to continue to build the pipeline behind it. And that's the exciting part, right? Is making sure it's you're still having the early research, and then you start also having programs that are knocking at doors of candidate selection, INDs, and looking at those things. And so that's what's coming for us in the next 2 years, and very excited about that journey for sure.
Absolutely, and I'm really rooting for you, because I think, like we talked about, the application of technologies, cancer is still a big problem, right? And we know that. And it's not been solved, and people are continuing to do great work on it. But there are other problems too, and I think applying some of those technologies more broadly is really key. And that's starting to happen, and you and some other companies are leading that. But I think obviously you have this sort of opportunity to treat the diseases you're working on, but it's also a sort of proof of concept for rolling out these tools from other areas into other things, and I think that's really important.
Yeah, well, thank you. Yeah, so we're really excited about what we're doing, of course, and we think it has great potential, right? So we're doing fibrotic disease now, but there are a bunch of other indications we're looking into, and so there is a lot of potential.
Yes. Well, we wish you the best of luck, Remi. Thank you so much for joining us.
Thanks so much, Tom. This was great. I appreciate it.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.